All right, in this video, we're going to do 10.3 to 10.5. It's not a lot of slides, so just make sure you follow along because there are more processes that we're going to talk about. So in 10.3, we're going to talk about how the Calvin cycle actually uses chemical energy of ATP and then also NADPH to reduce carbon dioxide to sugar. Remember, both of ATP and NADPH is being given to the Calvin cycle from the light reaction. So the Calvin cycle and citric acid cycle both have starting materials that they can remake once they get used up. So it kind of goes in like this circular process of just being used and then remade. The Calvin cycle is anabolic, so that means it builds things, from smaller molecules. So it builds sugar from smaller molecules specifically, and it uses energy from ATP in order to do it. And then it also uses an electron shuttle known as NADPH in order to carry those electrons, which get carried by the hydrogen. The citric acid cycle, on the other hand, is catabolic. So it oxidizes or loses electrons in order to get acetyl-CoA and to get the energy that it needs to make ATP. Okay, so what exactly happens when we are talking about the Calvin cycle? So we have carbon that's going to enter the Calvin cycle as carbon dioxide, and it's going to leave as sugar. And the specific name of the sugar that it leaves is called G3P, also known as glyceraldehyde 3-phosphate. When you have one G3P made, so when you're trying to make one G3P, you have to go through the cycle 3 times. And it uses 1 carbon dioxide per cycle or per turn. So to make 1 G3P, you need to have 3 turns of the Calvin cycle, and you use 1 carbon dioxide for every turn. Now, the Calvin cycle has 3 phases. It has a carbon fixation phase, it has a reduction phase, and it has a regeneration phase. And we are going to talk about those phases in the next slide. So this is the carbon fixation phase. So in the carbon fixation phase, which is phase 1, you're going to have CO2 that is going to combine with 5 carbon molecule. And when it combines with a 5 carbon molecule called Rubisco or RUBP, I'm sorry, not Rubisco, it's going to combine with RUBP. This is the first part. What's going to happen is this is going to make a 6 carbon compound that is super unstable. And it's going to split the 3 carbon compound called 3-PGA. And this reaction is going to be catalyzed all by a enzyme called Rubisco. So the 6 carbon that is super unstable will split into a 3 carbon compound called 3-PGA. Let me rewrite that again. And all of this is going to be catalyzed, like I said, by this enzyme Rubisco. So this is what happens in carbon fixation. So if you need to pause it and copy this down, you can do that now. Then we move on to the reduction phase, okay? In the reduction phase, you are going to have ATP and NADPH which is going to convert the 3-PCA that we saw was split into G3P. Okay? Once this happens, NADPH is going to reduce the 3-carbon intermediate to make the G3P by donating its electrons. So I'm going to write that down. So NADPH reduces the 3-carbon intermediate to make the G3P by donating its electrons. So this is reduction. Then in the regeneration phase, which is the third phase, you're going to have G3P molecules, not all of them, some of them, and they're going to make glucose. The rest of the G3P can be recycled. And they're going to make also a couple of the RUBP molecules. And in order for this to happen, you need to have ATP being used. And then RUBP will be ready to get more, will be ready to get CO2 in order for another turn of the cycle to occur again. So this is the third phase that happens. So that was 10.3. If you need to pause the video in order to go through it, please do that so that you can keep up with it. In 10.4, we're going to talk about plants that are in different environments and how they go through carbon fixation. So when plants came onto land, they started having problems with dehydration. Before, plants used to live underwater and they were pretty okay. But when they started moving onto land, they started having problems with dehydration. So plants have something called stomatas that are on the underside of their leaves that allow gas exchange as well as water exchange with their environment. So they allow CO2 to come in and oxygen to leave. When the plants are located in really hot environments and on hot days, plants have the ability to actually close their stomata so that they can retain the water and not lose it. But when they do this, it actually reduces their CO2 levels. Now, this is not a good thing for plants because plants do need CO2 in order to function. But the good thing, though, is that even though they have their stomatas closed to reserve the water and, they don't have a lot of CO2 levels, oxygen does keep being produced because light reaction is continuing to occur, and so is the breakdown of water. Now, this can lead to a wasteful process called photorespiration, okay, which we have right here. So there are ways that plants can protect themselves, but sometimes it can cause more harm than be an actual productive thing. So in normal plants, also known as C3 plants, you have carbon fixation with carbon dioxide by the use of the enzyme Rubisco that will actually help to catalyze this reaction. And this will form the 3-carbon compound that we saw earlier in the cycle. In photorespiration, you have the enzyme Rubisco, which will add oxygen, not carbon dioxide, and make a 2-compound molecule. It will take in oxygen and it will release carbon dioxide with no need of ATP or sugar being made. It uses ATP, but it will not make ATP, and neither will it make sugar. And this happens when there is light. People believe that photorespiration actually occurred when the atmosphere had made more carbon dioxide than oxygen. But photorespiration actually provides protection against damage from the light reactions, which can happen when you have a high amount of CO2. This process of photorespiration can actually be really harmful in plants, especially crop plants, because they take away more than 50% of carbon that is fixed by Calvin cycle. And that is a necessity for plants to function. And so you are not able to have the plants grow properly or function properly and produce whatever, fruit or vegetable they are trying to produce. And so it's not healthy. Now that we talked about C3 plants, we're going to talk about C4 plants. So C4 plants incorporate carbon dioxide into 4 carbon compounds, hence C4, to minimize the cost of photorespiration into the Calvin cycle. So C3 is 3 carbon compounds, C4 is 4 carbon compounds. These plants are efficient in hot and dry climates. So these are really good for those kind of areas. And they make a lot of energy. So sugar cane, pineapples, these are examples of plants that have this kind of process that happen. C4 plants actually have something called bundled sheet cells, which are pretty much tightly packed sheets that envelop each other around the veins of the leaf. And then they also have mesophyll shelves, which are loosely packed between the bundle sheath and the leaf surface. And those two make up these specific cells in the C4 plants. So sugar production in the C4 plants occurs in 3-step processes. The first step is that you have the mesophyll cells, the enzyme P. In mesophyll cells, you have an enzyme called PEP carboxylase, which will add carbon dioxide to the PEP, forming a 4-carbon compound. The PEP carboxylase has a higher affinity to carbon dioxide than the enzyme Rubisco. So it can fix carbon dioxide even when it is low. So remember, C3 plants have Rubisco as their enzyme to catalyze their reaction, but the PEP carboxylase in C4 plants are able to work and have a good proficient amount of work being done even when you have low carbon dioxide levels, which is really, really good and are beneficial to the environment that they live in. The 4 carbon compounds, for example, malate, they will move into a bundle sheet cell through a plasmodesmata, which we talked about earlier is this like super narrow, passageway of cytoplasm that passes through the cell walls, connecting each cells together with when the cells are next to each other. So it's like a secret tunnel between cells for communication, which is beneficial in this case. And so then in the bundle sheet cells, you have carbon dioxide that's going to be released and it will enter the Calvin cycle. Once it enters the Calvin cycle, you will have sugar that will be made. So this is kind of an image of what we just talked about. You have PEP carboxylase right here, which helps with the process of, creating malate, which will then go through plasmodesmata, which is like these little canals right here between each adjacent cell, and then it will go into the bundle sheath. Remember, it starts off in the mesophyll, enter the Calvin cycle to make sugar, and then it will go into the vascular tissue, which will transport sugar to other areas of the plant. Then there's something called CAM plants. CAM plants are plants that will actually close their stomata during the day in order to conserve the water, and then they will open them up at night in order to take up carbon dioxide when it's a lot cooler. They will combine the carbon dioxide and organic acids that they have in vacuoles, and then the mesophyll cells of the CAM plants will actually store these organic acids they make at night in vacuoles until the morning when the stomata closes, so that during the day, when light reactions can actually supply ATP and NADPH for Calvin cycle, this carbon dioxide can get released from the organic acids that were made at night and that were stored in the vacuoles in order for sugar production to continue in the chloroplast, which is really, really amazing that God would do such a thing and blows my mind every time I talk about this stuff. The CAM and the C4 plant pathways are very similar because they both put carbon dioxide into intermediate organic molecules before they enter the Calvin cycle. The difference is that in C4 pathway, structurally separate, you have structurally separate steps that are occurring between bundle sheets and mesophyll, where you have carbon fixation from the Calvin cycle. In CAM plants, the steps that are occurring for carbon fixation all occur in the mesophyll, but they occur at different times. So that's important for you to know, the difference between C4 pathway and CAM pathway. All right, last section. 10.5, life depends on photosynthesis. The importance of photosynthesis, basically a review. So remember, solar energy is converted into chemical energy. Chemical energy is stored in organic compounds. Sugar is made in the chloroplast, which provides the chemical energy and then also helps to make carbon skeletons in order to make organic molecules for the cells. Plants are able to store excess sugar and starch in chloroplasts. They can be later on secreted when they are needed. They can be stored in places such as roots and seeds and whatever, that has room for the storage. When we talked about light reactions, which we see here, they're carried out by molecules in the thylakoid membrane. So they're converting light energy into chemical energy of ATP and NADPH. So we see that here. Remember, they make ATP and NADPH that will go into the Calvin cycle. When they break water, it releases oxygen as a byproduct into the air, which is what we breathe in. And then the Calvin cycle, which is in the stroma, will take the ATP and NADPH from the light reaction to convert it to, it uses ATP and NADPH to convert carbon dioxide into G3P, and then it will return the ADP and NADP plus it used in order for a light reaction to convert it back into ATP and NADPH to go back into the Calvin cycle. And then it will make sugar as the energy source. We talked about photosynthesis. In chloroplast, photosynthesis uses light energy to convert carbon dioxide and water into organic molecules with oxygen as a byproduct. In mitochondria, we have cellular respiration that occurs, which breaks down organic molecules and stores energy in ATP. ATP powers the cell by active transport and protein synthesis, and then carbon dioxide and water are the byproducts. In the nucleus, in here, we can see that you have DNA gets replicated to make mRNA, which then goes into the cytoplasm. The mRNA attaches to a ribosome, which freely floats in the cytosol or cytoplasm. Or some of the ribosomes are attached to the rough endoplasmic reticulum, where proteins are made. And proteins are made by rough endoplasmic reticulum. They get sent to the Golgi apparatus to be further processed. You have transport vesicles that pinch off of the Golgi apparatus to be carried to wherever they're needed. Some of the proteins that are being made will merge with the plasma membrane of the cell and then release those proteins, while others will stay in the cell. Water can diffuse in and out of the cell easily through the plasma membrane through the process of facilitated diffusion. Passive transport, when carbon dioxide from photosynthesis comes into the cell and oxygen out of the cell. That's an example of it. In active transport, when you need energy to transport a solute against the concentration gradient. And then endocytosis and exocytosis move larger molecules into and out of the cell. So this is pretty much like what our cell goes through. And we can see how photosynthesis and the cellular respiration in mitochondria also help to make all of this, what I just talked about, actually occur. We need those energies being supplied by both of those important processes in order for any of these proteins and any of these things to be made. So in summary, light reactions are carried out by molecules in the thylakoid membrane, convert light energy, which is from the sun, into chemical energy of ATP and NADPH, and then they are able to split the water that releases oxygen into the atmosphere that we need to breathe. And then you have the Calvin cycle, which takes place in the stroma. Remember, Calvin cycle takes place in the stroma, while a light reaction takes place in the thylakoid membrane. In the Calvin cycle, you have ATP and NADPH from the light reaction that converts CO2 into sugar, specifically G3P. Then they will return the used ATP, which is now ADP, and the used NADPH, which is now NADP+, back to the light reaction so that the light reaction can convert it to ATP and NADPH and return it back to the Calvin cycle. So this is the end of chapter 10. Please make sure you work on your outlines. Write all of these processes in your outlines because they will be crucial for you to know for the quiz. There will be no quiz review, but you will need to know all of this information in order to answer the questions on the quiz.